The overall goal of this procedure is to demonstrate how to create a protocol that can analyze the cyanobacterial physiological state effectively, using flow cytometry and molecular probes.
This protocol can help answer key questions about the cell physiology of microbial communities. The main advantage of this procedure is that an optimized fluorescent probe protocol can distinguish between the physiological states of cyanobacteria at the individual cell level in real time. Demonstrating this procedure will be David Hartnell, a PhD student working in the Cytometry lab at Bournemouth University.
Before beginning the experiment, place an empty hemolysis tube onto the sample injection probe, click Unclog, and then backflush to start the flow cytometry cleaning process. At the end of the backflush, load a new hemolysis tube containing 2 milliliters of ultra-pure filtered water onto the sample injection probe, or SIP, and set the time limit to 10 to 15 minutes and the fluidic speed to fast. Next, select a new data cell and set the relevant fluorescence and light scatter thresholds to reduce the background noise. Then click Run. If the total events per second are not less than the manufacturer's recommendation, run 2 milliliters of decontamination solution for 2 minutes on fast, and repeat the backflush and ultra-pure water flushing steps. To prepare a primary aeruginosa monoculture, autoclave 98 milliliters of ultra-pure filtered water mixed with 2 milliliters of algal medium in a 250 milliliter beaker for 20 minutes at 120 degrees Celsius. When a culture is in a high steady